We have Miss Vijayashri. She is the senior scientist at Strand Life Sciences. She has been with Strand for over a decade. Her team has built one of the largest whole exome sequencing operations in the country, and she'll be walking us through that journey from early challenges to large scale success with real metrics and lessons learned along the way.
So with all the streamlining, this is the current whole exome sequencing workflow which we have in the lab currently. So we get major samples is whole blood which we receive from different hospitals across India. On receipt of samples, these samples are accessioned and barcoded and then taken forward into wet lab. So DNA extraction is the first step in the wet lab, which happens on automation Manta. So where the samples can be handled in a high throughput manner, and these samples are then followed with the QC. All the quantification and qualification of DNA samples are performed. All the qualified samples are taken forward in the library preparation, where the DNA undergoes fragmentation followed by ligation of adapters, followed by indexing, pooling, hybridization, and the qualified samples are taken forward into sequencing. The sequence samples are then fed into the strand NGS, where the variants are identified, interpreted, and the reports are generated. So just to touch upon with the QC steps in the workflow, what I have shown in the previous slide. So we are doing QC steps at different levels. Soon after DNA extraction, the DNA samples are quantified and qualified. So the samples extracted on Manta have given really a good yield, which is more than 3 microgram, and which is very much sufficient for further downstream assays. And with the whole exome, it requires just 25 nanogram of sample DNA. And then touching upon with the DNA quality, if you look at the when we checked on the protein contamination, A262 by 280 ratio have given really consistent value between 1.8 and 2, which is the expected value for any DNA to be of a good quality. The next QC step is followed post library preparation at pre-capture level, where we could see on the tape station the peaks are between 350 and between 350 and 450 base pairs, where it's a good quality library which can be taken forward for sequencing. So that is where whatever the samples extracted on Manta followed by library preparation have given a good quality library which can be taken forward for sequencing. Post sequencing, the samples are analyzed and interpreted, and the reports are generated, where it includes indication for test results and interpretation of summary, so which will tell if the variants are pathogenic or non-pathogenic, likely pathogenic, and it is according to the ACMG, American College of Medical Genetics. So it's a comprehensive report with interpretation and recommendations, which has been issued to the physician as well as for the patient.